So that was part one, and really an overview of what has been accomplished in 10 plus years, and it is a lot. Okay, part two is about IDG data resources. And over its 10 years, IDG produced a diverse array of data resources and discovery tools from a correspondingly diverse team of investigators and developers, merging expertise in foundational biology and medicine with informatics and computational methodology. The graphic here illustrates a sampling of these resources. From my own lab at Mount Sinai School of Medicine, there's GeneShot, DrugShot, Apaters, and ArchES4, and the Gene and Drug Landing Page Aggregator. The Clinical Kinase Index was developed by the Stephan Schurer Lab at the University of Miami. Prokino is a protein kinase ontology developed by the Kanan and Kochut labs at University of Georgia. Reactome IDG was developed by the Reactome investigators. Here at the University of New Mexico, we developed TINX, TIGA, and Drug Central, which Jamie Holmes will describe in a moment. And Pharos, the IDG data portal, was developed at NIH, NCATS, and will be described by Tim Shields in a few minutes. Another partial listing of IDG data resources includes a few more, and with links to additional information or the resources themselves. And yes, we will be sharing these slides in cooperation with the KC. So you can click and investigate more from this slide. Pharos, as the IDG data portal, integrates many of these resources for convenient and coherent access. So I tried to highlight those with the brighter or wider borders. Pharos in many ways sits on top and integrates many of the others. We don't have time in this webinar to describe all of these by any means and more IDG resources, but it is important to understand that all are designed to serve the same overall scientific use case, illuminating the understudied genome for potential new drug targets. And in a general but very accurate sense, providing evidence from heterogeneous and independent high-confidence sources to associate diseases or conditions with genes and proteins, which are hypothesized targets for novel pharmaceutical therapies, whether new chemical entities or repurposed drugs. So yes, there's a lot, but it's coherent and it serves a unified purpose in so many ways. Before turning it over to Jamie Holmes to describe Drug Central, I'll first briefly describe another featured and exemplar IDG data resource, TINX, Target Importance and Novelty Explorer. One reason for including TINX today is that there's a brand new publication just this week describing the latest version of TINX. The first author is Vincent Metzger, postdoc at UNM and project manager for TINX since 2021. The initial idea and prototype for TINX was developed by Christian Beloga of UNM. TINX briefly defines two statistics from bibliometric measures derived from the PubMed corpus via text mining, that is named entity recognition, and results from the lab of Lars Joel Jensen of the Novo Nordisk Center for Protein Research and the University of Copenhagen in Denmark. Novelty, one of those statistics, is a measure of the scarcity of mentions of a gene or disease, and importance measures the co-occurrence of a disease-gene pair, hence strengthening our belief in a Bayesian sense in that association or relationship. Via scatter plots and ranked lists, TINX provides literature-based hypotheses with provenance to the specific publications. The new publication about TINX includes a description of new data and features and examples of citations and usage by other investigators, indicating TINX's use and value.